look at cellular respiration. And with cellular respiration, you have the burning of glucose, the oxidation of glucose. And so you have some molecules that are being oxidized, other molecules that are being reduced in that particular process. So we're going to start by looking at oxidation and reduction and what that actually means in those particular cases. So both involve what we see as the movement of electrons. So the movement of electrons from one molecule to another. We typically call these a oxidation reduction reaction, or commonly redox. So redox just is an abbreviation for reduction oxidation, because one loss of electron is another's gain. So whenever you lose electrons, another molecule gains electrons. So there's conservation of energy. It goes back to the laws of thermodynamics again, and what we see is that law of conservation of energy. So what we call a loss of electrons, and this is a great term for you, is just an oxidation. So an oxidation is a loss of electrons. What we call the gain of electrons, or the addition of electrons, is a reduction. Okay, we call it a reduction. So when a molecule is oxidized, it loses one or more electrons. And when a molecule is reduced, it gains one or more electrons. And so let's take a look at this equation down here below. Here you have glucose, C6H12O6. And here you can see that you have oxygen. So it's being oxidized. The glucose is being oxidized in that particular process. You can see that we have 6 carbons here and we have 6 oxygens here. Okay, but in this particular process, what we produce is 6 carbon dioxides, and then we also produce 6 waters. So the electrons are being donated. The electrons are being donated here from the hydrogens to the oxygen, and the oxygen becomes the final electron acceptor. Okay, in this particular case, oxygen. Okay, this oxygen accepts the electrons and becomes water in that process. And we generate ATP, and we also give off heat. Okay, so heat's given off in that particular process. Okay, so here again, oxidation, you're losing electrons. Reduction, okay, you're gaining electrons, and so oxygen becomes the final electron acceptor in this particular case. Now it's important to note that you have 6 carbon dioxides, you also have 6 waters. That's the product of this particular reaction. So this is the reaction for cellular respiration. Glucose loses hydrogen atoms and becomes oxidized to carbon dioxide. Oxygen gains hydrogen atoms. In fact, you can see it right here. Okay, those are the hydrogen atoms that it gains, and it becomes reduced to water. Okay, now hydrogen may not seem like much, but keep in mind that hydrogen, right, has 1 proton, and circling it, it has 1 electron. So for every hydrogen, you have 1 proton and you have 1 electron. So 2 hydrogens, 2 electrons. Okay, 3 hydrogens, 3 electrons, and so forth. And when we take those electrons away here from glucose, we're giving them to oxygen. Oxygen becomes the final acceptor, and we're forming water in that particular process. A little tricky. So an important player in this transfer of electrons, is what is called a coenzyme, NAD, which actually stands for nicotine adenine nicotine adenine dinucleotide, NAD, but you don't have to remember that. You can remember it as NAD plus. Okay, NAD plus. In the plus in this particular case indicates that it's oxidized. Okay. In other words, it actually has one more positive charge than it does negative charges. So the plus, the 1 is assumed. So you could write it like NAD plus 1. Okay, but the 1 is assumed, so you don't have to write the 1 in there in that particular case. All right. So what does NAD do? Well, it's a carrier. It accepts the electrons, and it becomes reduced, forming NADH. So that NAD that we looked at earlier, plus, okay, when it gains an electron, all right, it becomes just NAD. And then when it gains a second electron, it becomes NADH. All right, so I'll show you what that looks like. Gains a second electron, becomes NAD. That forms a bond with the next hydrogen, so it becomes NADH. Okay, now you do have this lonely little hydrogen, H plus, because it's missing an electron. So there are two electrons here. There's one with this hydrogen forming a bond with that NAD, and then there's this one right here, which is just a proton. Okay, there's no electrons, so it has a plus 1 charge on that particular one. Okay, and you get two of these electrons in that particular process. One to neutralize the plus here, the oxidized NAD, and the other one right here to form that bond. So we call it NADH. But there's two electrons. So, backing up here for just a second. Every NAD carries 2 electrons. Okay, 2 electrons. And you can see those right here. They're symbolized right there. Okay. This is another example just kind of showing the same thing. When this particular molecule becomes oxidized, you're stripping away these 2 hydrogens right there, okay, and those electrons are forming a double bond, okay, right here. So that's a double bond that you see. Notice that there's a single bond to begin with. Remember, a single bond has 2 electrons here, right? But a double bond has 4. So you actually have 4 electrons here. Those extra 2 electrons are coming from these hydrogens right there. You might have to watch this a couple times to understand it in more detail. But let's take a look at this example down here below. So NAD plus in these 2 hydrogens, it's gaining 2 electrons in that process. So it uses one of those electrons to balance out this positive charge, uses the other one to bond, and forms what we see as NADH. Now it can give up those electrons, like you see here, and donate them in what's called the electron transport chain. And every time it donates these electrons, because they pass from here to here, and these are all channels, but every time it does that, it creates energy to make ATP. Finally, the last acceptor of those electrons is oxygen. And I said that before, oxygen's the final acceptor. And when it accepts those electrons, it binds with hydrogen to form water. So here we have 2 protons of hydrogen combining with a molecule of oxygen, actually half molecule, just to balance it out. But it forms water in that particular process. So the NADH delivers the electrons, and it becomes an electron transport, if you will, a carrier. These carrier molecules form what we see as the electron transport chain, but at the bottom of the hill, oxygens are acceptor. It accepts the 2 electrons, picks up 2 hydrogen ions and becomes reduced to form what we see as water ultimately. Okay, so water is given off. And just to recall, that's 6 waters in the process. Okay. So the steps that we see with cellular respiration, 3 main stages. The first stage is the breakdown of glucose. We call that glycolysis. So this is just the breakdown of glucose. Breakdown of glucose. Okay, it's called glycolysis. So glycogen, how glucose is stored up in animal cells, is broken down. This happens in the cytosol. We break down glucose into 2 molecules of a 3-carbon compound that we call pyruvate. Pyruvate. Okay. Then in the second step, pyruvate is oxidized, enters into what we see as the citric acid cycle as acetyl coenzyme A, which we'll talk about. But we oxidize pyruvate to this 2-carbon compound, which winds up being acetyl coenzyme A, okay, which we're going to talk about here in a moment. The cell makes a small amount of ATP during glycolysis. It actually makes a net here of 2 ATP. We'll talk about that too. Okay. It produces 4, uses 2, so the net would be 2 ATP in that particular process. But the big ATP generator is oxidative phosphorylation. Here again, we have NADH. We also have FADH, which is another coenzyme. Coenzyme term again. So FADH2 is another coenzyme. It shuttles electrons, both of them shuttle electrons through this electron transport chain, which is embedded in this inner mitochondrial membrane, which we're going to take a look at. The result of this is that energy is released as those electrons go downhill from NADH. They're donated from NADH and FADH. Energy is released to phosphorylate, to add, if you will, a phosphate group to ADP and produce in the process ATP, adenosine triphosphate, adenosine triphosphate. So as electrons pass through this chain, they move down this energy hill, and it pumps hydrogen ions across this inner mitochondrial membrane, and this produces a concentration gradient of hydrogen ions. And now as these hydrogen ions go down their concentration gradient from high to low, just like we've looked at, okay, it's a process called chemiosmosis, but that potential energy now from high to low in this concentration gradient is used to actually make ATP. So it's used to actually generate ATP in that particular process. All right. So let's take a look at the first step. This is kind of an overview. This is just showing you, here's glycolysis, glucose being converted to what we see as pyruvate. It's important to note that this is happening in the cytosol. So here's the membrane, the mitochondria, you can see right here. You can see these electrons are carried by these coenzymes NADH, FADH2, okay? Those are the reduced forms of those particular carriers, those coenzymes. Here you can see the citric acid cycle inside the mitochondria, and you can see along the way ATP is being produced. Most of it, though, is being produced here during oxidative phosphorylation as the electron transport chain and chemiosmosis generates the ATP. And that's what we're going to look at. So let's take a look at each one of these steps individually. Let's start with glycolysis.